So we're going to get back started early. We have the online crowd that's going to be joining us now. And it gives me a great pleasure to reintroduce you to Adam Jordan, who is working towards a Master's of Science in Food and Nutrition, as we heard about from his own research at Western University, and has also been a long-standing member of our Youth Advisory Council. So welcome back, Adam, for this session.
Thank you, Dr. Goldstein. So we're going to have a second rapid oral presentation, 3 more presentations. So we'll start with presenter number 1, Dana Soufi El Sabbagh, PhD. She is a 4th-year PhD candidate at the Hannan Ullah Lab at the University of Toronto, specializing in induced pluripotent stem cell models of bipolar disorder. Her research focuses on mitochondrial dysfunction in and neuroinflammation in bipolar disorder, leveraging iPSC-derived cerebral organoids to uncover disease-specific vulnerabilities in metabolism, neuronal activity, and inflammatory response. Welcome, Dana.
Hello, everyone. I'm Dana. Thank you again for having me here, and thank you to the CYBD committee for organizing this. Today, I'd like to talk to you about a project we have at the Hannan Ullah Lab involving unraveling mitochondrial, inflammatory, and neuronal vulnerabilities in bipolar disorder using iPSC-derived cerebral organoids. I have no conflicts or disclosures. I don't need to give you a background on bipolar disorder for today's audience. However, the motivation behind this research is that bipolar disorder is a debilitating disease. Patients go through high relapse rates. They have a reduced life expectancy, and unfortunately, current treatment options are very limited. At the Hannan Ullah Lab, we like to have a patient-specific approach. We use patient-induced pluripotent stem cells from blood cells of patients with bipolar disorder, and from there, we generate a number of neuronal cells to really investigate the bipolar disorder vulnerabilities within these neuronal models. We also specialize in looking at the mitochondria. A little bit of background on the mitochondria. It is not just the powerhouse of our cells. It is a double membrane-bound organelle in charge of ATP production through oxidative phosphorylation. It regulates a number of key aspects in our body, including redox balance, calcium homeostasis, metabolic pathways, as well as immune system signaling. The mitochondria has its own DNA, known as the mitochondrial DNA, mtDNA, is maternally inherited and unfortunately is very highly susceptible to damage from reactive oxygen species, or ROS. Now, oxidative stress can lead to mitochondrial dysfunction, and this leads to dysregulated cellular bioenergetics, dysregulated ATP production, and the release of damage-associated molecular pathogens and free radicals. When the mitochondria is released from the cells upon stress, we refer to it as circulating cell-free mitochondrial DNA, or CCF mtDNA. And this can contribute to neurodegenerative, metabolic, and inflammatory diseases. So beyond mood dysregulation, mitochondrial dysfunction is highly implicated in bipolar disorder. Patients with bipolar disorder have shown to exhibit high levels of lactate as well as CCF mtDNA. And we've previously reported on a large cohort of 837 patients with BD where we found high CCF mtDNA indicating some sort of chronic low-grade inflammation within these patients. Another important aspect to look at within neuroinflammation in BD is the NLRP3 inflammasome and how sensitive patients with bipolar disorder are to activation of the NLRP3, which is a nod-like pyrin domain tripartite complex in charge of releasing pro-inflammatory cytokines in the body. And so we want to explore the bidirectional relationship today between mitochondria and inflammation in bipolar disorder using disease-specific models. So our study comprised of 3 non-psychiatric healthy controls and 3 patients with bipolar disorder. We take the patient's PBMCs, peripheral blood mononuclear cells, we electroporate them and generate induced pluripotent stem cells, or iPSCs, and from there we can differentiate these stem cells into a variety of cell types like neural progenitor cells, cortical neurons, or in our case for today's work, 3D cerebral organoids. For those of you who are not as familiar with 3D cerebral organoids, or COs, They are a three-dimensional tissue structure comprised of neural progenitor cells, neurons, and astrocytes. They mimic some brain-like cytoarchitecture as they have these ventricles of the 3D cerebral organoids. This is a cryosection of one at a 1 month time point where they contain mostly neural progenitor cells. And as these organoids progress for long-term culture, 4 months, 5 months onwards, these neural progenitor cells further differentiate into neurons and astrocytes. So for today's work, for today's project I'll be presenting, we are going to be looking at these 3D cerebral organoids in patients with bipolar disorder and investigating their mitochondrial profile as well as their inflammatory profile. So a little bit of a summary on how we generate these organoids. First, we culture our iPSCs in 6 well plates. We then singularize these iPSCs and take them into an EB, embryo body formation, where they form little clumps, almost like a tiny three-dimensional clump of cells. We then allow them to grow in neural induction media to allow for the ectodermal growth differentiation, embed them in matrigel, and allow for the three-dimensional spheroid shape to form, which forms a cerebral organoid. Typically at day 27 or at 1 month is when we look for the SOX2 neural progenitor cells. SOX2 staining for neural progenitors, GFAP for astrocytes, and UN for postmitotic neurons. Now, with these organoids, we wanted to look at a number of mitochondrial endpoints. First of all, we wanted to look at the overall organoid health, measuring how many cells we have per organoid in the control and the BD groups. We found significantly reduced number of cells in the bipolar organoids. We then went on to look at ATP production, normalized to cell number, in the control and the BD. And again, we found a decreased ATP concentration in the BD group in comparison to control, almost a 50% decrease. So this was telling us there is some sort of energy dysregulation and imbalance in these bipolar organoids we generated. We then went on and ran a metabolomic profile across the disease groups. We've heard a little bit about metabolomics today through some talks and some poster presentations. Metabolic health is very important in bipolar disorder. And what we found interestingly here was decreased levels of glucose 6-phosphate, which is the first breakdown after glucose in the glycolysis, as well as decreased levels of ATP, again, in the cerebral organoids of the BD group, indicating there is some sort of dysregulated energy metabolism in these organoids. Some more specific mitochondrial endpoints, we did some live imaging dyes using MitoTracker Red. MitoTracker Red stains polarized healthy mitochondria. And we quantified this and found significantly lower number of polarized mitochondria in the BD organoids compared to the control. We then stained for MitoTracker Green, which looks at mitochondrial mass. It stains mitochondria regardless of health. And we found similar levels of mitochondria across the disease groups, indicating although control and BD might have similar levels of mitochondria, not all the BD mitochondria are as functional as the control. Now, we allowed these organoids to mature up to 6 months in order to allow these neural progenitor cells we saw at the first month to further differentiate into mature neurons, staining for MAP2, and astrocytes, staining under GFAP. And then we looked at some electrophysiological activity to look at, are these neurons firing, are they functional, and is there a difference between the disease groups? Again, we found increased number of spikes in the BD group. We also found a severely decreased number of duration for these spikes. So there was some aberrant electrophysiological activity indicating dysregulated neuronal firing in the BD group in comparison to the control when the organoids matured. Now, we've talked about the mitochondrial aspect as well as the overall organoid health. In terms of the inflammation, we wanted to also interrogate that profile and how sensitive these organoids are to inflammasome sensitivity and chronic low-grade inflammation. We measured levels of CCF mtDNA, a marker of oxidative stress and mitochondrial stress, and we found those to be elevated in the BD group. And so from there, we went on to investigate NLRP3 activation sensitivity, since this inflammasome activation has been known to be increasingly activated in mood disorders, including bipolar disorder. For those not as familiar with the NLRP3 inflammasome, it is a NOD-like receptor domain. It's a tripartite complex made up of the NLRP3 protein, ASC spec, and caspase-1. And upon activation, it produces a lot of pro-inflammatory cytokines like caspase-1, IL-1 beta, etc. We can activate the NLRP3 in our cells through a two-step process. First, by priming toll-like receptors using LPS, lipid polysaccharides. This initiates NF-kappa beta transcription, translation, and then by activation through nigericin, a bacterial toxin, which then initiates the NLRP3 complex to form together, shown in the purple, where you see ASC, caspase 1, NEX7, and NLRP3. And this forms the NLRP3 inflammasome. And we can actually visualize this in our cells using immunofluorescence microscopy. So I have baseline and activated here. Baseline indicates no NLRP3 activation. Activated indicates we treated with LPS and nigericin to activate the complex. And what you see here is GFAP for astrocyte staining and ASC to show us ASC speck formation. These ASC are tiny dots shown in purple in the images, and they only form when the NLRP3 complex is activated. So we can tell whether it's activated through these immunofluorescence images and by quantifying them. So we did this in the control and the BD to look at NLRP3 activation at baseline across both groups, and upon activation to see how sensitive each disease group is to this activation. And this graph is a little bit complicated, but the blue bars represent baseline levels. Between control and BD, there's no significant difference at baseline in terms of NLRP3 activation. However, upon treatment with LPS and nigericin, or activating the complex, we see significant increase in both groups. However, we see an even more significant increase, almost 130% more, in the BD group in comparison to the control, indicating the BD organoids are much more prone to NLRP3 activation and sensitivity compared to the control. We also wanted to try inhibiting the inflammasome complex as a drug treatment. We know that MCC 950 is a classical NLRP3 inhibitor, and we've also previously collaborated with a group in Brazil that uses BFE, a bioactive flavonoid extract, to inhibit NLRP3 activation. And so we wanted to test the treatment of both of these pretreatments on control and BD to see if we can inhibit activation of this complex. So what we did here was we primed the system, gave it a pretreatment of either MCC 950 or BFE, and then we gave nigericin to see if the system will be activated. And upon quantification, the first graph on the left shows us MCC 950 giving almost 100 to 110% rescue effect, indicating NLRP3 activation was decreased completely. And the second graph in purple shows the BFE treatment, indicating, again, BFE did rescue the system from NLRP3 activation by almost 80 to 100%, a little bit stronger in the control group in comparison to the BD. Now, this project is currently being finalized as a paper. It's currently under review, and it's also posted on bioRxiv. For those of you who are interested, you can scan the QR code and take a deeper look at it. But from what we can tell from this is these bipolar disorder cerebral organoids exhibit disease-specific phenotypes. We saw some nice evidence of mitochondrial dysfunction, increased inflammasome sensitivity, and this dysregulated neuronal firing. And this brings us to the conclusion of cerebral organoids are a nice novel platform to really dissect these BD-associated cellular mechanisms and look at the different molecular findings and clinical interventions using this platform. I'd like to thank my lab at the Andreazza Lab as well as collaborators. Thank you very much.
Thank you, Dana. Very interesting work, and those pictures were really nice. So do we have any questions? Yeah, over there.
Hi there. Beautiful presentation.
Thank you.
And thanks for such a nice, amazing overview of cerebral organoids. My question, I guess, is you're seeing a lot of effects here, and I'm just curious for your thoughts on how distinct do you think they are? Because when I'm thinking about altered bioenergetics and the cell are the electrophysiology results that you showed, how do you know that the inflammation is not just like a carryover of altered excitability of the mitochondria? Like, just maybe help me understand that a bit more.
You're asking if the inflammation is not an effect of mitochondrial dysfunction?
That's right, yeah.
So we don't. We actually think there might be chronic underlying inflammation that could be causing the mitochondrial dysfunction, or it could be the mitochondrial dysfunction causing this increased inflammation. It's almost like an intertwined bidirectional relationship, because with dysfunctional mitochondria, you have elevated levels of CCF mtDNA being released into the periphery, and then that itself can actually induce inflammasome activation and allow for increased inflammation in the body. So it's almost like a feedback loop.
Okay, thanks.
I actually had one question, Dana. That was an awesome talk. In terms of the 3 patients with BD and then the 3 controls that you selected, did you have any specific kind of criteria for the participants you selected? Like, was it just 3 general sampling? And then for your controls, like how healthy were your controls? Were they allowed to have other kind of medical conditions or risk factors that can also influence mitochondrial health?
At the time of recruitment, I believe these patients this is with a collaborative study in Australia at Deakin University. This was done for the controls. They're non-psychiatric healthy controls. At the time of recruitment, we did not know of any other conditions. And I have the clinical data on hand. I can show it with you later, but not any known diseases with the healthy controls. For the bipolar, originally it was 5 patients that we selected, but when you generate stem cells, not all of them pass karyotyping and characterization, so we ended up with 3. And it's a general subset. I believe 2 of them have bipolar 1 has bipolar 2, and one of them is also a non-lithium responder. So it wasn't a very rigorous criteria, but I can definitely share the more specific details of that.
Thank you.
I'm wondering what your thoughts are about mitochondrial genetic variants and how they relate, or how important that is, and what proportion of the phenotype that you're presenting do you think is genetically driven?
That's actually something we're working on right now. Mitochondrial genetic variants play a huge role in bipolar disorder and mood disorders in general, and we're actually working on some patient subsets now, looking at patients with bipolar disorder and a family history of mitochondrial disease versus patients with just bipolar disorder. Mitochondrial DNA can definitely influence things like energy metabolism, ATP production, and neuronal signaling. And that's something we're looking to further investigate in the future, both with these samples and with additional samples. Yeah.
I have a couple of online questions. The first is, so interesting, thank you. Are there any implications for everyday nutrition at this point to assist with mitochondrial function? So pairing the last couple of presentations.
I'm not a dietitian or a clinician, so I can't speak too much on this, but in terms of, you said nutrition, right, and mitochondrial function, there has been a lot of recent studies on the ketogenic diet being very beneficial to patients with bipolar disorder, as it has helped patients regulate their manic and psychosis episodes a little bit more. Metabolism is a very big thing in mood disorders and bipolar disorders specifically, so definitely huge implications there. That's all I can say, as I'm not a registered dietitian, to comment more.
Okay, thanks. And then the other question is, this relates to diabetes and sex differences. Is the common factor explaining the observed association with diabetes, assuming the diagnosis is valid? And what is the overlap with mTOR signaling, known as a factor which is elevated more in men than women?
I'm not quite sure about diabetes. I don't think any of the patients we have in this study have been diagnosed with diabetes, and I know that the 3 control and bipolar are age and sex matched, and they're actually all female. So I don't have an answer to that right now.
I'll add one more just because we're good for time. Can you just make explicit for folks what exactly is involved in terms of time and attention to create these things?
So yes, for sure. When we look at clinical studies, there's always such a huge N number, and then when we look at studies involving stem cells and organoids, the N number is much smaller, and that's because the time it takes to generate stem cells to begin with from a patient blood can take up 6 to 8 months, and that's if it's succeeded from the first try. Like I said, sometimes your karyotyping can fail, and then you would have to redo it again. And then once you have your stem cells, you need to culture them for 20 passages or 20 weeks if they grow at a decent timeline, and only then are they considered mature enough stem cells to start generating cerebral organoids. So that's already a couple months before you even start the organoid generation. For organoids, it does take a month to generate a 1-month organoid containing of neural progenitor cells. But when you want to look at things like more mature neurons and astrocytes and look at inflammatory signaling, you need to wait 6 months for these organoids to mature. And it's very time-consuming, it's very costly, and obviously these are all cultured without antibiotics to mimic a physiologically relevant environment. So you really want to hope for, you want to have the technical skills, be very specific, proper aseptic cell culture techniques, and a lot of patience. So typically about a year if you want to go from patient blood to an organoid. Yeah. Thank you.
Thank you, Dana, and thank you for all the questions. So our next presenter is Hannan Ullah. He is a second-year MSc student at U of T's Institute of Medical Science. He is currently working under the supervision of Dr. Stefan Kloiber at CAMH and is co-supervised by Dr. Jacob Vorstman from Sick Kids Research Institute. Hannan completed his Bachelor of Medical Science from Western University in 2022 and previously worked with Reach Nexus at St. Michael's Hospital as part of the Our Health Box research program. His thesis work focuses on the use of smartphone-derived data to detect early signs and symptoms preceding mood episodes in young individuals living with bipolar disorder. Welcome, Hannan.
Thanks so much, Adam, and it's going to be a tough presentation to follow. There was a great job that was done previously, so I'll do my best. And thank you so much for this opportunity to be able to speak today. So today I'll be presenting on a study that I'm overseeing as part of my thesis for my master's entitled Detecting Mood Episodes and Symptoms in Young Individuals with Bipolar Disorder Using Automatically Generated Smartphone Data. And just before I begin, I want to give a quick acknowledgement to my co-supervisors, Dr. Kloiber and Dr. Vorstman, as well as my committee members not listed on the slide, Dr. Li Shu and Dr. Goldstein. And again, I don't have a slide to show this, but I have no conflicts of interest to disclose. And I know I'm preaching to the choir a bit, but just to kind of underscore the importance of this study, we know that bipolar disorder has a heightened proportion in terms of who it affects in the youth population, so those aged 15 to 24. And we know that a higher proportion of youth individuals living with bipolar disorder will suffer a new mood episode within a year after recovery of a first episode. And with an increase in the frequency and severity of recurrent mood episodes, there are higher societal costs associated with social and occupational disruptions, as well as progressive cognitive deterioration, otherwise known as neuroprogression. And we can see that in an alarmingly increased risk of suicide. And perhaps one of the key factors in combating this is to identify what are called prodromes. So these are subclinical behavioral changes that occur prior to a mood episode. And the prodromes that have been relatively well described in terms of episodes of mania are, of course, increased energy, pressure speech, meddling and arguing, and distractibility in a couple of weeks to a month and a half preceding an episode of mania. And in terms of a depressive episode, we can see decrease in speech, lowered energy, and slowness of movement. But even though these are well described, they're by no means homogeneous. Of course, everyone's experience is quite different. And oftentimes, these prodromes aren't recalled prior to an episode as they're happening. They're only recalled during a full mood, when a mood episode is in full swing, or after the episode has remitted. Now, with this, of course, this impedes the possibility of early intervention and prevention, which are currently limited in clinical settings. And so the main challenge is how can we objectively recognize prodromal symptoms in real time? And so in the past 10 to 15 years or so, a field known as digital phenotyping has emerged. So this essentially refers to using active and passive data from smartphones and wearable devices to provide low burden near continuous data. And the subset of digital phenotyping there's a subset of digital phenotyping known as passive monitoring. So this is the collection of smartphone data in real life settings without active participant input. One app that enables this is called BHAP, which collects info about physical activity and mobility, social communication, phone usage, and even sleep-wake data. And the app has been previously shown to be able to detect prodromes in individuals with schizophrenia. And so with our study, we want to see to what extent can personal smartphone data derived from BHAP be used to detect behavioral changes that occur early in the development of a new mood episode in young individuals diagnosed with bipolar disorder. And in terms of this, the study, we initiated a prospective observational longitudinal study wherein we recruited individuals living with bipolar disorder between the ages of 13 to 45. I will address that in a moment. Who've experienced a mood episode, either mania, hypomania, or depression within the past 2 years or at the time of inclusion. And because the app only works on Android or iOS smartphone, we do require them to own one of those. And we would exclude individuals experiencing active symptoms of psychosis that are unrelated to a mood episode or have a current diagnosis of a moderate or severe substance or alcohol use disorder. And so we installed the app on the participants' smartphones and followed them for 12 months. During this time, they completed monthly self-report questionnaires. They completed the PHQ-9 for depressive symptoms and the Altman self-rating mania scale for manic symptoms. And of course, we followed up to confirm any mood episodes using the SCID-5 according to DSM-5 criteria. So our mandatory follow-up visits were at 6 months and 12 months. But if we received a notification about elevation of mood symptoms based on the self-report data, we would also carry out a follow-up visit then. And so just in terms of results, right now we still are in preliminary stages. We've recruited about 31 individuals, but we've only analyzed about 22 just because of the sheer time it takes to generate a data summary. And so far, 11 individuals have completed the study. And in terms of the analysis I'll be presenting, again, it is quite preliminary at this stage just to explore and investigate what potential features are related to mood symptoms and mood episodes. So we can see that smartphone and app usage is associated with both depressive and manic symptoms, such that lower usage of the smartphone is predicted by an increase in depressive symptoms, as we can see by total time spent in communication apps, the number of times apps were open per day, and the number of times phones were unlocked. So this survived both unadjusted analysis and analysis adjusted for age and sex. We also saw an increase in depressive symptoms that predicted reduced activity, but not manic symptoms as much. So we saw that an increase in depressive symptoms predicted fewer daily steps and greater home stay, which is consistent with other studies in this field. And we saw that an increase in manic symptoms predicted an increase in the distance traveled when participants were moving, but this was only in the unadjusted analysis. And in terms of differences between actual mood episodes, we saw a lower duration being spent still or stationary, and a greater number of trajectories in individuals in a euthymic state versus a depressive state. And we observed a higher number of stay points and a higher number of times that the smartphone was unlocked in individuals in a euthymic state versus a depressive state. And my apologies, I forgot to mention that these results are derived from a linear mixed effects regression model, which essentially is designed for individuals enrolled in a longitudinal study. So we we set the smartphone data, the smartphone-derived behavioral features as the dependent variable, and set the mood symptoms and mood states as the independent variable, and added time and day of the week as fixed effects. And we accounted for each individual participant's behavior from day to day as a random effect, just to account for that variation. And so in terms of conclusions and future directions, it is a bit reckless to generalize the results at the moment, but they are pretty consistent with the research that's been done with respect to wearables and smartphone passive monitoring. But we do want to move on, before moving on to the next steps, we are hoping to kind of refine the feature engineering. So in a sense, getting a better sense of, okay, which features derived through BHAP are potentially redundant, which ones are reliably detecting changes in mood symptoms and mood states through processes like structural equation modeling and principal factor analysis and principal component analysis and so on. And of course, the next step would be, and following that would be to create a machine learning model and a smartphone instrument that we could potentially test and also look at on the individualized, on the individual level. And so ultimately, our goal is to have an instrument that could objectively identify prodromes in individuals, which could overcome challenges in episode recognition and facilitate early intervention. And this would be integrated with mobile interventions such as CBT, DBT, and IPT, and also be integrated with care management. Thank you so much.
Thank you, Hannan. That was very interesting, and I've never even heard that word prodromes before. Yeah, so that's learned a lot there. Do we have any questions? I guess I could ask one. So it showed the, when there's the instance of the prodromes happening, and then you kind of go down into whatever the next state would be. Do you ever see sort of a false flag there, where you kind of feel like it's going, and then you're good?
Absolutely. So that absolutely can happen. Of course, like within our study, there are quite, like as you saw on the slide, there's a low number of mood episodes that happen. So it could happen that, okay, as your mood is going down, potentially take medication or seek out an intervention that prevents that from happening. Sometimes, individuals just have a more mild or less severe phenotype of bipolar disorder there may be a drop in mood or a slight increase in mood, but it won't develop into a full mood episode. So I can't conjecture too much on like why that happens or how often that happens, but absolutely. So essentially that's why it's important not to, again, jump straight from the data we have into applying it in a clinical setting.
Thank you for that talk. Very interesting and very easy to collect that data. I'm wondering what you're hoping for in terms of the future of this data. So say you are able to very quickly predict an upcoming mood episode. What do you hope will happen if your smartphone detects an upcoming one?
Yeah, so, as mentioned, the hope is to kind of facilitate early intervention and prevention. Which is like, as I also mentioned, is not as well established as it should be, given the consequences associated with bipolar disorder. So of course, because we are a ways away, we are hoping for like a smartphone tool that would be developed, and then we are in the process of potentially integrating other features such as speech sampling. But yes, kind of moving toward a new model of care to facilitate more virtual strategies for prevention and intervention.
Thanks for your talk. Does it matter what they're doing on their phones? I mean, what if somebody's up all night playing video games versus somebody who's actually interacting with other people? Is there a way to discriminate those?
Yeah, so we're collecting, so thank you for the question. There's about 104 different features that are collected, and so those are all categorized into app usage, activity, mobility, and communication. So with the example that you provided, we do see different categories of apps that are being used. So communications, entertainment, news and magazine, health and fitness. So that kind of gives us an idea of how much time is being spent on the apps. Of course, for ethical and privacy reasons, we don't see exactly what they're doing. We just know that it's the duration for which it's been open on a daily, weekly, and monthly basis, or the number of times it's been opened. And there's some other standardized metrics that were tested, but didn't turn out to be significant at the moment.
Yeah, can I just say, I think this is a really important development in the area, the potential to use technology in this way. In our own work, rather than looking for sort of group differences, we're kind of preoccupied with the individual trajectories that people are on, the variation within, and therefore the extent to which people will be able to use that to understand their own trajectories and what is happening as the ways in which people relapse or the thresholds are quite different. So I just wonder, in the data you've already got, have you been looking more, not just for the group effects, but for the nature of the individual trajectories that I think in the end of the day will be things that will be able to be taken back into clinical practice?
For sure, yeah. So I think one of the secondary or one of the additional primary aims of the study is to look at those differences on an individual participant level. At the moment, we are kind of in the process of developing that, so that will come as we're developing you could say like classification or machine learning analysis, and then develop those individual participant or patient profiles. So that's definitely in the pipeline. Thank you.
You may have mentioned this, but did you control, like if you're looking at A to B associations, did you control for mood symptoms in that model, predicting? Like, so looking at the associations of incoming phone calls, activity, that sort of thing, with depression symptoms, could you just reiterate the model?
Yeah, so the model we used was a linear mixed effects regression model. So we set the behavioral features. So for example, I'll just go back here. Like number of steps, home stay, distance travel, number of incoming calls, outgoing calls as the dependent variable, and then the symptoms as a first level of analysis, the level of symptoms as a predictor. And then we also, and then the next phase of analysis, we set the mood state as a predictor. And then we added day, so time as a fixed effect just to account for that. And as we also added a random effect on the level of the participant just to account for individual variations in behavior.
Thanks.
All right.
Awesome.
Thank you.
Thank you.
All right. So our third presenter is Joyce Jiang. Joyce Jiang recently completed her PhD in the Department of Pharmacology at University of Toronto. Her PhD research was conducted at the Center for Youth Bipolar Disorder, supervised by Dr. Benjamin Goldstein. She led work focused on polygenic risk score for bipolar disorder and demonstrated that adult bipolar disorder polygenic risk is similar among youth, pushing back against the stigma regarding the diagnosis in youth. Her thesis also focused on intermediate phenotypes in youth with bipolar disorder and included neuroimaging and cognitive analyses. Welcome, Joyce.
Thank you, Adam. All right, hello everyone. My name is Joyce, and as Adam just mentioned, I recently finished my PhD at Center for Youth Bipolar Disorder. So for today, I would like to share with you some of the findings included in my thesis titled Polygenic Risk Scores and Intermediate Phenotypes in Youth Bipolar Disorder. I have no disclosure. So bipolar disorder is one of the most terrible psychiatric disorders with a heritability of 60 to 85%. And compared to adults with bipolar disorder, early onset bipolar disorder is characterized by a more severe course of illness and is also found to be more familial, which highlights the importance of genetic factors in youth bipolar disorder. Recent findings from genome-wide association studies, or GWAS, have suggested that bipolar disorder is highly polygenic. And the most recent and the largest GWAS, just published in 2005, identified almost 300 genome-wide significant single nucleotide polymorphisms, or SNPs, but each SNP only has a very small effect size, no larger than 1.1. So in order to capture this cumulative genetic risk to a polygenic disease, polygenic risk score, or PRS, has been developed based on the results of GWAS. PRS can be generated in each individual in the study cohort, also called the target sample, and is calculated as the sum of the number of risk alleles carried by individuals weighted by the effect size of each risk allele. And the risk alleles and their effect sizes are derived from the GWAS summary statistics, also called the base data. PRS, therefore, models aggregate index of a genetic liability to a polygenic disease in each individual in the study cohort. And the more genetic risk alleles that individuals carry, the higher score they will get, which means higher genetic risk for that disease. So over the course of my PhD, we examined the polygenic underpinnings of clinical neuroimaging and neurocognitive phenotypes in youth bipolar disorder. And overall, we hope that this line of research may help provide biological validation of youth bipolar disorder to help reduce doubt and stigma and to eventually improve early identification and risk stratification. So the first thing we were interested in was the specificity of bipolar PRS that is derived from adult genetic data in relation to bipolar diagnoses in youth. Specifically, we look at PRS for 4 psychiatric disorders, including bipolar disorder, schizophrenia, major depressive disorder, and ADHD. And we found that there is a similar trend with all 4 psychiatric PRS across groups, but the only significant difference was for bipolar PRS, where youth with bipolar disorder, as shown in orange, has significantly higher bipolar PRS when compared to healthy controls, as shown in green. And bipolar PRS among youth who were at high clinical or familial risk for bipolar disorder was insignificantly in between. And PRS for schizophrenia, major depressive disorder, and ADHD were not significantly different across groups. We then examined bipolar PRS in relation to several intermediate brain-based phenotypes, including gray matter structure, white matter integrity, resting state function connectivity, and neurocognition. So starting with the neurostructural matrix, we found a similar pattern of negative associations with bipolar PRS, where higher bipolar PRS was associated with smaller gray matter and white matter matrix, regardless of diagnoses, particularly in regions involved in higher order cognitive functions and also processing and regulation of reward, emotion, and visual information. So specifically in this figure, the x-axis is bipolar PRS and y-axis is the standardized values of significantly associated brain matrix. And the bipolar group is shown in orange and the healthy control group is shown in green. Just want to note that we also did a similar analysis in the combined sample of youth with bipolar disorder and healthy controls and found similar associations. So specifically within the bipolar group, higher bipolar PRS was associated with smaller cortical thickness of frontal, temporal, and fusiform gyrus. And within the healthy control group, it was associated with smaller cortical volume and surface area of the superior frontal gyrus. Moving on to white matter integrity, we only found significant findings in the combined sample of youth with bipolar disorder and healthy controls, where higher bipolar PRS was associated with poorer white matter integrity in 7 clusters within the projection fibers that connect cortical regions and the brainstem. And because the associations are very similar for all 7 clusters, I'm only showing 3 here for illustration purposes. As for the resting state functional connectivity, in the oral sample, we found that higher bipolar PRS was associated with lower connectivity between the salience network, which is involved in the detection and integration of emotional and sensory stimuli, and left supramarginal gyrus, which is a key region within the salience network. We also found that higher bipolar PRS was associated with higher connectivity between the frontal parietal network, which is involved in the goal-directed cognitive tasks, and left precentral gyrus, which is a key region within the motor control network. Within the bipolar group, higher bipolar PRS was associated with lower connectivity between the visual network and left frontal pole, which is involved in higher order cognitive processes, and there was no significant findings within the healthy control group after correction for multiple comparisons. Finally, we examined bipolar PRS in relation to neurocognition, also in the oral sample, and then separately within the bipolar group and within the healthy control group. And we found similar negative associations regardless of diagnoses. Specifically, within the bipolar group, higher bipolar PRS was associated with poorer sustained attention and decision-making, and within the healthy control group, it was associated with poorer affective processing. So in summary, we found that bipolar PRS was significantly and specifically elevated in youth with bipolar disorder when compared to youth at high risk for bipolar disorder and healthy controls. We also found that higher bipolar PRS was associated with smaller gray matter matrix in frontal and temporal regions, poorer white matter integrity in projection tracts, altered resting state functional connectivity of the salience, frontal parietal, and visual networks, and finally, poorer neurocognitive performance on affective processing, decision-making, and sustained attention. So why does all of these matter? Well, despite all of the research progress over the past 20 years, there are still ongoing controversies over the validity of youth bipolar disorder. Therefore, demonstrating that youth bipolar disorder has a similar genetic risk structure as adult bipolar disorder is an important advance to reduce misconceptions and stigma over youth bipolar disorder. Our findings provide preliminary evidence that even with adult-based GWAS, PRS still shows similar predictive performance in our youth bipolar sample and demonstrated strong links with intermediate neurophenotypes. So these genetic findings add on to the fact that youth bipolar disorder shares a similar clinical course of illness as adult bipolar disorder, and together add further evidence supporting the validity of youth bipolar disorder. So with that, I'd like to thank patients, research participants, and their families, and our funding sources, and special thanks to our collaborators who have made this thesis possible. Thank you for listening.
Thank you, Joyce. Definitely some very important work. Any questions over here?
Thanks, it was really interesting. This is kind of a statistical question. If you put diagnosis in first as a predictor, does BD-PRS predict above and beyond diagnosis? And say in predicting gray matter or white matter?
Yeah, we did a control for the bipolar diagnosis in our analysis, and the findings remain similar. So I think bipolar PRS did provide complementary and separate explanatory power over the diagnosis itself.
Is it a lot of variance accounted for above and beyond diagnosis?
I actually haven't looked at that. So for our study, we only report the variance explained by the bipolar PRS itself. In relation to diagnoses, the variance explained by the bipolar PRS is around 8%, and in the neuroimaging model, it's a little bit higher, around 20%. I haven't looked at how much more you gain by adding, separately adding bipolar PRS versus bipolar diagnoses, but that's definitely something I could check later. Yeah, thanks for the suggestion.
Just in terms of your first slide about the PRS scores, just to understand, make sure I haven't got this wrong, you're talking about you had 3 groups of people, the young bipolars, the at-risk, and the others, and then you looked at the PRS scores for their schizophrenia PRS, for their MDD PRS, and for that, that one, So they're the PRS groups. I mean, like much work in this area, I mean, what's striking is, in fact, the similarity across the areas. Okay, that the high PRS for all those disorders across the thing. So this kind of shared genetic kind of idea, the idea that there are PRSs independently for all these things, often when you look at patient groups, you see exactly this, is this shared kind of group. So I wonder then in relation to your brain imaging and other findings, how your group sort of sees that. What's the shared neurodevelopmental kind of architecture really that's playing out here, that's operating across disorders?
Yeah, that's actually definitely something we will be interested in looking at for our next step. So for my thesis, we'll only look at bipolar PRS specifically in relation to these neuroimaging phenotypes, just because in this first project, the bipolar PRS is the only one that's significantly and specifically different across groups. That's why we only targeted this PRS in the following neuroimaging and neurocognition projects. But in the literature, a lot of people have looked at PRS for other psychiatric disorders in relation to neuroimaging phenotypes in both clinical groups and general population. And there are a lot of findings that is overlapping with our current findings. So, for example, for the PRS for schizophrenia, which is the most correlated PRS with our bipolar PRS, a lot of studies reported similar associations in frontal and temporal regions, which we know in the prior non-genetic, like purely neuroimaging studies, we also see a lot of common findings in frontal and temporal regions and neurocognitive domains similarly reported among, kind of shared among all these psychiatric disorders. So definitely for our next step, we would be interested to look at PRS for these other psychiatric disorders in relation to the intermediate phenotypes.
So I guess the other way around is everyone's trying to understand what are the neurodevelopmental things that are actually shared in common that lead to disorders, and then how is it that given these sort of shared origins, we end up with the different disorders that we see. So in terms of exploring that further, does your group have particular kind of views about that? I mean, people are always trying to sort out, is there anything specific that is not just the shared common element?
Yeah, so our, regarding the neurodevelopmental aspect, our cohort is only cross-sectional, so we don't really have longitudinal data on the specific neuroimaging phenotypes. And we haven't really looked at how a bipolar sample can be distinct with, for example, a sample of patients with major depressive disorder. But I know there are a lot of studies going on that's either look at purely neuroimaging phenotypes and how it can be distinct among different psychiatric disorders, or looking at PRS for a specific psychiatric disorder, so non-shared. So people pick out the non-shared risk alleles to calculate, for example, a bipolar specific PRS, and use that PRS to examine the additional neuroimaging and cognitive correlates. I see the literature right now is not very consistent. I haven't seen very compelling or consistent findings yet, but I know this is definitely a pretty hot topic going on. And maybe we know that with the larger sample of GWAS and the more advanced statistical tool to calculate PRS, they can be more and more specific and explain more and more variance in diagnoses and neuroimaging phenotypes. So definitely looking forward to see if the field can be more, can discover more specific phenotypes that's unique for each psychiatric diagnosis or what's shared among diagnoses. Yeah.
All right. Thank you, Joyce. Okay, thank you for all the presenters for the rapid oral presentations. Here's Dr. Goldstein. Give a hand for Dr. Goldstein.
Thanks, Adam. That was great. I thought that these, you guys hosting these sessions worked out perfectly, and a lot of great questions. So this brings us to the end of our formal activities today. I just wanted to do two things. One is to call folks up who are going to give awards to our speakers, or gift bags, I should say, and also to encourage folks to come back next year, to encourage other people to come. You know, our focus, as I mentioned, is bipolar disorder, youth mental health, physical health, connections with psychiatric illness. And also to plug that tonight we're going to be having we have two events. Typically we used to have a solely public-focused patient and family evening, and over time we started this research symposium. But if you're around, either virtually or on site, come to our evening event tonight. So it starts online at 6:30 and doors open here at 6 p.m. We will have a fantastic presentation from Amelia, who's here today, who's going to speak about her lived experience, and then from some early career investigators, myself, and then collaborators with the Mood Disorders Association of Ontario. So maybe a little bit less focus on research, a little bit more clinical, and also more patient lived experiences. So consider that online. It's on the same Eventbrite. You can do it, or just let one of us know on the way out that you plan to come back. We'd be happy to have you. Same room, same place. So thank you for joining, and we'll call you guys up. And this is Golda, for everyone who heard the name Golda 10 times. Dr. Miklowitz. Thank you for joining us from Los Angeles and great talk. Dr. Hickey, who stepped up at the last minute. Surprise, this one is Dr. Vasilev. Sunny. Kristen? Last but not least, we have Adam. Maybe final comment to say that if you have ideas, I'll do this for people online too. If you have ideas about topics you'd like to see covered, about speakers that you'd like to see invited, not to say that we can always necessarily follow them, but we're open to it and would love to expand this network. So thanks for making this, I think, a really meaningful day. I can't separate whether it's the greater attendance, the better room, or whatever it was, but I think this was our best year yet. So come back for year 5. Thank you.